Lunchtime.
Biotechnology has created powerful weapons against disease, but these have not translated to cures. Because many drugs are delivered as unstructured mixtures that fail to reach the right cells at the right time. Flashpoint Therapeutics ends this gamble through a revolutionary nanotechnology platform that precisely controls drug components, structure, and timing. This technology has shown promise against debilitating cancers in humans. It has transformed ineffective drugs into highly potent treatments in models of breast, lung, skin, prostate, brain, cervical cancer, and lymphoma. Flashpoint will bring the same benefit to patients by building a pipeline of next-generation drugs for multiple cancers and other diseases. The bold Saudi 2030 vision has materialized in bringing this revolutionary approach first to Saudi Arabia. In partnership with inventor Chad Merkin, winner of this year's King Faisal Prize, and internationally renowned executive and cancer biologist Adam Margolin as CEO. The first drugs will be tested and brought to patients in the world-class hospitals in Saudi Arabia. This revolutionary nanomedicine, born from Saudi vision and leadership, will save lives around the world.
Adam, you can speak to the panel. Yes, to the panel.
Yes, okay. Imagine that you are locked in a room and there's a ticking bomb on the other side of an uncrossable chasm. So in desperation, you just throw wire cutters at it and hope that they just happen to land on the right combination of wires at exactly the right time. You can think of cancer as that ticking bomb in our body, and biotechnology has created powerful weapons against it, but they've not translated to cures because, like defusing a bomb, defeating cancer requires precise, coordinated interventions. But drugs today are delivered as unstructured mixtures that cannot control where they go or when they activate. Flashpoint overcomes these limitations by transforming unstructured mixtures into precise nanostructures that precisely control the combination of components that are co-delivered into cells in optimized structure and timing of activation, and delivers these exactly into the right target cells. This overcomes the key limitations that have prevented biotechnology advancements from translating to cures, and develops a next-generation therapeutic approach that can finally bridge that chasm. As evidence of that, for a nanotechnology immunotherapy delivered to mice, we observe that 35 times more of the components are delivered into cells. We achieve 70 times more potent activation of anti-cancer immune response, and we achieve 350% increased killing of tumor cells. This all amounts to that therapies that are ineffective when delivered in their conventional form are transformed into cures. Not just for one example, but in fact for all 9 mouse tumor models that we've tested to date, the same components that are ineffective in their conventional form are transformed into highly effective immunotherapies, at times reaching up to complete cures. And this approach brings hope even to patients suffering with debilitating, life-threatening cancers. A study performed by a previous group before Flashpoint has shown that even patients with life-threatening, incurable terminal metastatic cancer was cured by nanostructure technology. From the business side,
time is out.
From the business side, the approaches currently in this sector have given rise to multiple multi-billion-dollar companies, but these have not translated to curative therapies because they all cannot overcome this chasm of key technological limitations. Flashpoint overcomes all of these because we're developing a new, rationally designed nanotherapeutic that's unlike anything that has come before.
Thank you, Adam.
Okay.
If the panel has any questions for Adam, Your Excellency, Lubna.
Well, I will leave to the esteemed
Okay, yeah.
the technical question. I just want to vote for the mission. I think cancer obviously is, as indicated in the beginning, a ticking bomb. It's a big scare. It keeps many individuals and families awake at night worried about what is going to get them. So I think as we have people working on longevity, people working on quality of life, reducing the cost of healthcare. These kind of innovations and solutions, by all means, tick all of the boxes, whether you think about it at the individual, family, even at the corporate level. When I was CEO of Saudi Aramco, and we looked at what are the things constraining the company, healthcare and productivity of our employees, including the incidence, the high incidence of some types of cancer, was one of the issues we discussed on the board and around the management committee table. Surgery and long-term cures are costly, so something like this gets me excited, and once again I would vote for it. When I heard about this, I talked to our Saudi FDA and they called me back. They said, thank you very much for sending them our way. We will do an accelerated clinical trial introduction into the Saudi population.
Thank you your support on this, and they will do their clinical trial in Saudi because of the help that you are
Exactly.
providing. Thank you.
Adam, thank you. Very good presentation. I'll leave the technology part and the medical part to all the experts here. Let me ask you, who are your competitors and what gives you the edge? And also, quick question, you've done this on mice now. When are you ready to go to human? So how far are you from there? And what do you need other than money from the health sector?
Great questions. Okay, so competitors. So on the previous slide, there's two primary sectors that we can compare to: cancer immunotherapies or vaccines, and then drug delivery in general. The competitors are listed on the top. Moderna, BioNTech are some of the big ones, Alnylam and so forth in drug delivery. They're using technologies primarily based on lipid nanoparticles, as well as other ones. Our technology overcomes many of the limitations that they have faced, primarily in terms of accumulation in liver, not being able to be delivered to target cells, not being able to co-deliver different combinations of therapies. So we listed the matrix up here and why we overcome many of the key limitations that they've faced. In terms of milestones, other great question that's coming up right here. So first of all, all-star team from across top universities across the country. Milestones, seed round led by Beta Lab gets us to our first clinical candidate being ready for clinical trials. We will have a clinical candidate ready within 1 year, and then it's depending if we can do an accelerated clinical trial in Saudi, we can do it then. And then phase 2 is a broader pipeline. We'll have multiple phase 1 and 2 clinical trials, which will be actually ahead of the competitive companies that at the time of any of their IPOs.
We need one more comment or question before we move to the next one.
So let me stay very focused, right? You've got the IP essentially to be able to take an effective molecule and target a very specific cell type. What gives you confidence? I mean, cancers are notorious for figuring out how to get around the therapy, right? And that's recurrence. How are you thinking about the right combination so that you minimize the chance of recurrence? I'm always cautious when you say cure, right? Cure over what time frame? Cancers do come back.
Yeah, so the one example that we showed was complete cure long-term to the extent that they followed the patient, no sign of recurrence. So that was one example. Of course, that doesn't happen all the time. The beauty of the platform is that we can actually target all of the escape mechanisms because we can co-deliver multiple components in the same structure. So we can deliver molecules that target the escape routes, that target the drivers initially, such that when the cancer overcomes one of them, we'll have other weapons that are co-delivered at the same time. What we're doing now and how we optimize that is exactly by testing those different combinations of components and then looking for which ones have long-term durability in the preclinical models and then doing that in clinical trials. That's what we're doing for our first candidate. That's the work over the next year, and then in 1 year we'll have an optimized multi-component construct that will be ready to target multiple of those escape mechanisms.
And
Thank you, Adam.
Are you at the seed round or the Series A round?
We just closed our seed round with Beta Lab.
Thank you, Adam.